Okay, we have been talking about cell signaling, and we have seen that the cell signaling more behaves like a black box. There is a signal coming in, and there is an output, a behavior, and we don't really know what is going on inside. There is loss and loss of molecular interactions, loss of proteins are bumping to other proteins and changing them, and altogether at the end they give an output. They change something. Okay, within these molecular interactions, there is something that we call molecular switches. So unlike the rest of the interactions, they have two states, like these old-fashioned lamps, on or off. So either you turn on the signaling or you turn off the signaling, and they are very useful that way because they sharply start a behavior. And these molecular switches work by phosphorylation. Yep. And we know that there are two resources for phosphorylation, either ATP or GTP. Now, for ATP and GTP, they are nucleotides, and we know that they are the resources of energy obtained by the oxidation of glucose. They are also the monomers of the RNA. They are also messengers. They are involved in signaling. And here, they are also used to turn on and turn off the molecular switches. So one of the molecular switches is, it works by ATP. So, in this case, when the ATP turns on the interaction, ATP joins and ADP leaves, and this phosphate group, the terminal phosphate, the triphosphate of one of the three phosphates, is incorporated into the enzyme or the signaling molecule, whatever. So as long as this phosphate is there, it is going to be active. So, of course, it cannot stay active forever. At one point it has to inactivate. Otherwise, a signal being active for too long can be toxic, can be dangerous. It has to be turned off, and to turn off, you have to remove the phosphate group. So it turns back to the initial state where it is inactive. So this kind of signaling molecules, they shuttle between the active and the inactive states by adding the phosphate from the ATP and by removing the phosphate group from the ATP. Of course, the added phosphate here is organic because it's coming from the ATP, and the removed phosphate here is inorganic because now it's just the phosphate group, inorganic phosphate group. Now, there is the off state, on state, it goes between them, and the enzyme that turns it on, which is adding the phosphate, is called the kinase. I'm sure you're familiar with this enzyme. We have been seeing the kinases since the first year. It adds the phosphate group, and the kinases are so fundamental, we see them everywhere. And in contrary, the enzyme that is removing the phosphate group is called the phosphatase. Okay, last time I realized that some of you thinks that the phosphatases add the phosphate. No, it's not. Please do not confuse that. The phosphatase removes the phosphate, and it usually turns off the signal. Yeah, that's it. And we have many of them. We are going to see the main kinases and the main phosphatases. Okay, so there are two main kinds of kinases, I can say. One of them is the serine-threonine kinase, and the other one of them is the tyrosine kinase. Now, what is common between all these three amino acids is that they all have this hydroxyl group. The phosphate is added to this hydroxyl group. This is why you cannot phosphorylate alanine. There is no alanine phosphorylation. There is no alanine kinase. Similarly, there is no, let's say, threonine is there. There is no glycine phosphorylation. Glycine doesn't have such a hydroxyl group that you can add the phosphate group to. So serine and threonine, their structure is quite similar. just threonine is one carbon larger than that. So whenever there is a serine or threonine, the kinase can add the phosphate groups. And the tyrosine has a cyclic group, a benzene group, but at the end it has this hydroxyl group also. This is why the proteins having these two amino acids can be phosphorylated. I can say that generally the kinases are more selective, and the phosphatases are kind of more generic. Up. We don't have much phosphatases, though we have several kinases in the cell. This is the story of kinases. We will always come back to them. Kinases are so central. The second group of molecules which are regulated by phosphorylation is the GTP binding proteins, also called GTPase. Now, we have seen examples of them. In cell transport, there was a monomeric GTPase called RAP protein, RAP GTPase. It was involved on the surface of the vesicle, and it determines or it helps the tethering protein which organelle the vesicle is going to dock. So we also see the GTPases everywhere. Now the main difference between the ATPases, and these are the ATPase, and these are the GTPase, is that when the phosphate group is added to the ATP using enzyme, only the phosphate group is added. In case of GTPases, not the phosphate group, but the entire GTP is added. So when it is interacting with the GTP, now I'm talking about the entire molecule, it is active. And when it removes the inorganic phosphate group, PI, when it ends up with the GDP, which has two phosphate groups, it becomes inactive. And it shuttles between these two states. Now, there are monomeric GTPases and trimeric GTPases. We have seen one of the monomeric GTPases, RAP GTPase on the vesicles. And we are going to see the trimeric GTPases today in chapter 16, which are also called GPCRs, a very famous class of molecules. Now, the enzymes, this interaction with the GTP, of course, is non-covalent. Unlike here when the phosphate group interacts with the enzyme, it is covalent. This is why adding and removing it requires enzymatic activity. But here it is non-covalent. Although it's non-covalent, it also requires enzyme activity to remove it. Now, okay, let's start from here. When there is GTP, it can be hydrolyzed, and this hydrolysis is induced by GAP, GTPase activating protein. So this PI is removed and it becomes inactive. And in this case, in the other one, the GAF cannot simply add the PI, because we know that this inorganic phosphate cannot be added here. It can only be added in the Krebs cycle. Here too, the phosphate group cannot be easily added to ADP, to make ATP. It can only happen in mitochondria using the energy of the glucose. Because you cannot make GTP out of GDP, what you should do is just remove it. Just take it off. Just get rid of it. And get another GTP from the environment. I mean, there are lots of GTPs and ATPs around, and there is an affinity for GTP. As you remove the GDP from the inactive monomeric GTPase, a GTP can join easily. And the molecule which removes the GDP is called GAF, guanine nucleotide exchange factor. So guanine nucleotide exchange factor activates the monomeric GTPase, and GAP, which is the GTPase activated protein, it inactivates the GTPase. So this way it goes and comes back between the two states. Okay, 9 minutes, let's stop here.